foods are products which contain an ingredient or ingredients whose DNA has been specifically altered. Presence of these modifications can be detected using a technique called the polymerase chain reaction. Genetic modification of foods has been a controversial issue due to debated concerns over health and environmental safety. The ability to detect genetically altered DNA in food samples of interest allows for educated decision-making about the safety and potential dangers of using genetically modified organisms, or GMOs, in food supplies. The polymerase chain reaction, or PCR, is used to amplify food DNA to determine the presence or absence of genetically modified sequences. Gel electrophoresis then pulls the amplified DNA through an agarose gel matrix and separates DNA bands of different sizes that correspond to modified or non-modified markers. These are then compared to control bands from foods known to contain GMOs or known to be modification-free. This video will illustrate the principles behind detecting genetically modified DNA from food samples, how to extract DNA and amplify markers used in the genetic modification process, and how the presence or absence of GMOs in food samples can be established. Polymerase chain reaction identifies sequences of DNA that have been inserted into the genetically modified food. DNA is a relatively stable molecule, so viable DNA fragments suitable for amplification can be isolated even from highly processed goods, like corn chips or vegetable burgers. A small number of regulatory DNA sequences are used to control the expression of inserted genes, so these sequences are present in the majority of GM crops. This procedure identifies two of the most commonly used, the 35S promoter gene and the nopalene synthase terminator gene. The 35S sequence is a strong promoter, and when attached to an inserted gene will drive constant high levels of expression. The nopalene synthase terminator is included to stop transcription of the inserted gene at the desired endpoint. PCR involves repetitive heating and cooling cycles in a thermal cycler, a machine that tightly controls the temperature of PCR reaction tubes. Heating the sample to 94 degrees causes DNA strands to denature and separate. Rapid cooling to between 45 and 65 degrees allows primers to anneal to the separated DNA strands. Finally, reheating to 72 degrees allows the Taq polymerase enzyme to extend the primers and complete duplication of the target region. Purchased plant primer is added to each sample and will amplify in samples containing plant DNA. GMO-positive templates for 35S and NOS are used to provide a positive control for GMOs. A certified non-GMO is used as a negative control. If any of these control reactions show unexpected bands, the results of test samples cannot be trusted. Amplified DNA is run through an agarose gel by electrophoresis. PCR products are mixed with a dye and loaded into wells. DNA is negatively charged, and when loaded into the gel at the cathode end of the chamber, will move toward the anode end when current is applied. Larger DNA fragments cannot move as easily through the gel matrix, so will stay closer to the cathode, while smaller sequences move toward the anode end, resulting in separation of differently sized DNA into distinct bands. After electrophoresis, gel staining helps visualize separated DNA bands. Now that we are familiar with the principles behind GMO detection and the use of PCR and electrophoresis for GMO identification, let's take a look at how this can be carried out in the laboratory. Once the food products of interest have been identified, analysis can begin. To extract the DNA, take 2 clean screw cap tubes, and into each of these, transfer 500 microliters of purchased DNA isolation reagent. making sure to pipette the mix up and down between aliquots to evenly mix the reagent. Label one of the tubes non-GMO and the other test. Next, weigh out 0.5 grams of certified non-GMO food.